I think it's time. So welcome everyone to the University of Washington Data Science Seminar. I am delighted today to introduce Professor Yee Yeung today to you. Yee is a professor in the School of Engineering and Technology at the University of Washington, Tacoma, and an adjunct professor in the Department of Microbiology at the University of Washington, Seattle. Her research focuses on the development of methods and software tools that leverage cloud and container technology that allow biomedical researchers to build modular, reproducible bioinformatics workflows. As usual, please ask your questions in the Q&A box. I think Yee is gonna divide her presentation in two parts, and she's ready to take your questions at the end of the first part. For the first part of her presentation, and the second round of questions at the end of the second part of the presentation. So thank you for joining us today, Yee, and please.
Thank you for the introduction, and also thank you for the invitation to speak at the UW Data Science Seminar today. So it's great to have this virtual interaction with all of you. So please feel free to ask questions. So I will try to try my best to answer your questions. So before I could go anywhere with science, I am required to disclose my financial interest. And let me know if you have any questions and concerns about the potential conflict of interest. I would be happy to provide more information if you would like. And I would also like to say that most of the work presented today are covered in a US patent that was submitted last year as well. Now to the science. Modern day biology is very much a data-rich science. There are so many different massive amounts of data have been generated to help us to advance our understanding of biological mechanisms. So in this talk, we would focus mostly on let me see if the cursor works, sequence data and gene expression data. But there are many different data types out there. For example, imaging data, proteomics level data, electronic medical records, and also text mining as well. So our overarching goal is to make latest technological advances accessible to biomedical scientists by developing easy to use tools to facilitate analysis of many different types of big data. So some of the characteristics of our tools is we use software containers extensively to enhance reproducibility. Our tools are modular so that the users can mix and match, and our platform are extensible. And we also use the cloud to help us with the big data analysis. And one of our goals is to optimize our tools to make them more scalable and efficient. And we will talk more about each of these characteristics later in this presentation. So our projects are called BioDepot, and our GitHub repo is /BioDepot. So feel free to visit our website, and we would appreciate your feedback and suggestions. And I would like to acknowledge my colleagues in the middle of the talk, not at the end. Ling Hong Hong is a research scientist in my lab, and he has been instrumental in the development of methods and tools presented today. Dr. Wes Lloyd is a faculty member also at UW Tacoma. His expertise is in cloud computing and software engineering. So there are two main themes of projects in my group. So first of all, we're going to talk about the methods and tools we develop to facilitate reproducible research. And next, the second theme is we are going to talk about the types of optimization we do to improve the performance of computationally intensive tasks. So first of all, before we talk about the type of tools we we develop, I would like to give a little bit more background in terms of bioinformatics data analysis. So bioinformatics analysis usually involve a pipeline or workflow consisting of a sequence of computational tasks. So, or we call each of these tasks a module. This is why we want to make our tools modular, because in a sequence of steps, so that the user would be able to do some mix and match. If they want to swap out one module, would be able to use a different method for this particular step. And each of these tools, each of these tasks in a workflow would have its own computational characteristics, have different resource utilization characteristics, and also could require a different computing environment and have different software dependencies or versioning required to run the tool, and could require different configuration or different ways to deploy these each task in the workflow. And another characteristic of bioinformatics workflows is because bioinformatics is a very interdisciplinary area, so that we work extensively with biomedical scientists. So graphical user interfaces are essential to facilitate collaboration and to facilitate interpretation of results. So here are a list of some of the methods and tools we developed in the past few years to facilitate reproducible research. So we started out with a GUI doc project, a GUI, graphical user interface. So the idea is to add a GUI on top of Docker containers. So in our first attempt, our first efforts, we used X11 to add a common graphics user interface to containers. And then in the next paper, we call our framework GUI doc VNC. We use no VNC to add a graphical desktop sharing system to provide a browser-based interface for containerized software. So the idea is that the user would be able to go to any browser and then connect to the port that we have opened up in the Docker container, and would be able to interact with the containers without using command line tools. And we also use Jupyter notebooks extensively. And in bioinformatics, bioconductor and R are used extensively for statistical analysis. So we created an app called BioC Image Builder that automate the creation of Docker files and Docker images to enhance reproducibility of data science notebooks. So this is this work. And oh, by the way, all these photos are students who are key contributors to these projects. And we are going to spend more time to talk about the BioDepot-workflow-builder project today. So before we talk about the BioDepot-workflow-builder project, I would like to give some more background on the types of workflows that we are going to use in this presentation today, or to illustrate our methods and tools. So we are going to talk about RNA sequencing, RNA-seq data and workflow in this presentation. So a typical RNA-seq workflow consists of three major steps. The first step is the alignment, in which reads are mapped to the reference transcriptome. RNA-seq is a widely used next generation sequencing tool. So the data are, the reads are essentially short character strings. And so in the first step, we are trying to map these reads, these short character strings, onto the long reference character string there. So that this is typically the most computationally intensive step in the workflow. And after the alignment step, the next step would be the table of counts. The idea here is to quantify the number of reads associated with each transcript. So you might think of the table of counts as a spreadsheet. Each row is a transcript. And in human, we have roughly 20,000 transcripts. So one column would be one RNA-seq experiment. So typically in RNA-seq experiments, we have multiple experiments. For example, we might have 3 experiments profiling the expression level, the activity level of transcripts in the normal, not diseased, like in control groups of patients. And then we might have another 3 experiments that profile the activity levels of transcripts in a deceased state of patients. So that we would think about would be a table. We will have 6 columns, 3 normal, 3 deceased, and then we have 20,000 columns for each of the transcripts in human. And then the next step would be differential expression. The goal here is to identify a list of differentially expressed genes across the different experimental conditions. So in the example I just talked about, the conditions would be normal versus diseased. So we would try to find among all the 20,000 transcripts, we want to find a smaller list that show different patterns between normal versus diseased state. So what we would do next is we are going to discuss some of the challenges associated with a workflow, such as the RNA-seq workflow I just described. And then over this talk, we are going to present different methods and tools that will address each of these challenges. So one of the challenges is that each step in the workflow requires a different computing environment and have different resource requirements needed. For example, let's go back on the previous slide. So in the alignment step, it's typically the most computationally intensive. And then this step, the tools are typically written in C, C++. Typically some compiled form of languages. And then in the differential expression step would be typically we would use R and bioconductor tools to perform this step. And the second challenge would be, as I said before, it could be computationally intensive, especially the alignment step. And yet another challenge is that cloud computing has been often used for big data analysis. But however, using cloud computing platforms typically requires substantial technical expertise, and that could be a bottleneck for interdisciplinary collaboration. So in the first project, we are going to present a graphical front end that will allow the creation of containerized modules without writing code. So BioDepot-workflow-builder, our acronym is BWB. This work was published in Cell Systems in 2019. So what we are seeing here is the graphical abstract from the paper, from the Cell Systems paper. So what we're going to talk about is some of the features in the tool BWB. So BWB offers a drag and drop user interface. So hopefully you can see it on your screen. So we have each of these icons is what we call a widget. Each of it represents a computational task or a module that is in the workflow. So a recap is a workflow has multiple modules and multiple tasks. So the user would be able to drag these widgets into the canvas. And by connecting these different widgets, we'll be able to create a workflow in an interactive and graphical manner. And we support interactive execution and customization of workflows for non-programmers. So we have a form-based user interface that the user would be able to change the parameters without changing the code. And we also allow users to import his or her code and to create containers without writing any code. So we have a user-based interface to support all of that. And our tools, we also have tools to facilitate integration of new modules and workflows into BWB. So you could add another widget by importing existing code that you already have, or to just add a container in the workflow. And everything in the tools is a container. So each of these widgets we talked about is a container, and BWB is distributed as a container of containers, so that you can run it anywhere that Docker is supported. So you can run it on your local host, your laptop, your desktop, or you could run it on the cloud. and we support a graphical output. So, one of our features is that windows can be popped up to support the interdisciplinary collaboration that we have been talking about. And so integrating graphical modules is easy. For example, we have Jupyter integrated in BWB as well. And I know I have already used the word interactive before, but I want to emphasize the importance of interactive analysis. This is the key to help to facilitate interpretation of results, that you could easily change the input parameters in all steps in your workflow. At the same time, if like in your lab, if 10 more samples of data got generated this week, you would be able to, with a simple click of a button, to rerun the analysis and update all your results and visualization as well. So a little bit more details on the widgets. So a widget is a modular unit in a workflow. So such as an executable, script, or basic operation. Each widget essentially calls a container in the backend, and a widget can be dragged to the canvas and connected to form workflows. So this is a visualization, an example, RNA-seq workflow. In this particular RNA-seq workflow, it uses a widely used workflow called Callisto Sleuth. So each of these step is a module and a widget. And the workflow is when the pipeline is to be run, the BWB engine would follow the graph, executing each widget and propagate the output. So for example, after this widget finishes running, the output of this widget would become the input to the next widget. And so all you need to do is to click a start button here. And then with at the end, when the workflow is done, a spreadsheet would pop up to show you the differentially expressed genes in this case. So to run BWB, all you need is to download the latest Docker image, and then you can run use a Docker run to start the container. And you can access the container by opening up a browser window and type localhost followed by the port number. You can open up any port number here. So this is our GitHub page, and we have very comprehensive documentation as well as a user manual, troubleshooting, and YouTube videos. So here, I would like to give you a quick demo here. So in this window, this is the Callisto Sleuth workflow. But in this example, unlike the one I showed in the slides, this one has the Jupyter integration in there. So when you run the workflow, I think it doesn't, I think my network is slow. So you click the start of the button. I'm not going to click it again, but you can click the start of the button and then the workflow will run. And the workflow encapsulates all the steps, including the data download, getting the reference, and then this is the index, and the Callisto quant is the creating the table of counts, and then the differential expression step is performed in the Jupyter notebook. So at the end, the Jupyter notebook would pop up. In the interest of time, it's like a cooking show. I have pre-cooked a little bit, and now I'm pulling out the notebook. And the user will be able to interact with the analysis and to change parameters and rerun the notebook again. So at this point, I would like to pause and take some questions. Let me see if I could find the chat box. Here, Q&A. Should I just go to the Q&A box, or is there a different box in the chat box?
The Q&A box is the one with the questions. Please put your questions there for Kari.
I don't see any questions, so maybe I would continue, and I would come back to check the Q&A box again at the end. Thank you. So I would come back to the presentation here. Here, so we talked about the first theme of our projects, and now we are going to talk about the second theme, which is holistic optimization of computationally intensive tasks. So here, I would like to talk about what do we mean by holistic optimization. There has been a lot of very nice methods, optimized methods, especially for the alignment step. But in our group, what is unique is that we optimize a workflow in many different ways. We optimize the algorithm, we optimize the software implementation, we also optimize how the workflow is being, how different each task is being deployed. And we also optimize the system's architecture on the cloud. And by accounting for many different types of optimization, we hope to get to further improve performance. So before we present the tool, just like last time, I will talk about a little bit more about the case study that we'll use to illustrate our methods and tools. So in this particular data case, in this particular case, we use a data set in data and workflow from the Drug Toxicity Signature Generation Center from Mount Sinai in New York. So in this project, so our collaborators, their goal is to look at the drug-induced adverse events caused by drug perturbations. So they profile the gene expression level, the activity levels of different transcripts in response to different drug perturbations. So they use cell lines in these experiments, and they look at 3 different types of treatment. The first group experiments are the control. The second group of experiments are a group of 24 drugs they call the NIPs, which are the protein kinase inhibitors that can cause cardiotoxicity. In the third group, these are 17 drugs that they call the non-NIPs. These are non-cardiotoxic drugs that are frequently taken by patients along with the NIPs. So they have developed their own in-house workflow to process these UMI RNA-seq data. So I should add a little bit detail. So in addition to the RNA-seq experiments, they use unique molecular identifiers to tag the 3' end of each of these transcripts for sequencing. The goal of that is they could make it do more experiments at a lower cost. So in this presentation, we are not going to focus on the experimental details, but we are going to more focus on the computational workflow. So this is very much a workflow that mirrors the general framework that, that we have previously discussed. So first of all, in the alignment step, they use a particular tool called BWA, the Bureau's Wheeler Aligner, to align short sequence reads against a reference sequence. We're going to talk a little bit more details of the BWA in a bit. And the next step would be the table of counts. And then they perform more quality control, removing outliers, do some data normalization. And the differential expression step is performed using a bioconductor package called HR. And then at the end of the day, we would get a list of differentially expressed genes that show different patterns of activities between the control and the drug perturbations. So BWA, it's a tool that was developed by Lee and Durbin in 2009. So the goal here is to align the short sequencing reads against a large reference sequence, allowing mismatches and gaps. So one of the issue is that it takes a while to run. It's computationally intensive. So the original workflow given to and the original standard operating protocol and all the code provided to us from our collaborators at Mount Sinai, mostly written in Python. And for 600 to process 640 million reads, which is roughly 46 gig of FASTQ files, take 29 and a half hours on a EC2 instance, an M4 4x large EC2 instance. And due to the substantial time requirement for the alignment step, typically the alignment step is rarely repeated, even though parameter choices could greatly influence the quality of the alignment. In their Barroso et al.'s 2017 paper, they used simulated data to demonstrate how changing the alignment input parameters can really change the quality of the alignment results. So here, what we are going to do is before we present our tools and methods, we want to showcase to illustrate the importance of improving performance. So improving performance, saving execution time is always a good thing. It saves us time, it saves us cost on the cloud, because typically the cost of running analysis is directly proportional to how long you need to run the job. But at the same time, what we want to show here is we want to demonstrate changing the alignment step parameters can actually change the biological conclusions that will be drawn. And by improving the runtime performance, by making these analysis instead of 29.5 hours to minutes, that would allow the user to change the input parameters many times, to tune all the parameters and the workflow. And that would actually subsequently could change the interpretations of the results. So here, what we are looking at is the changing one particular input parameter in the BWA step. The input parameter is the seed length. A seed is a short sequence. So you can think about a short character string. So typically, many alignment tools find these seeds matches on the reference to speed up the work. So the idea is to find to match these seeds, a short character string on the reference, and then could match the short reads by extending the seed, allowing for mismatches, insertions, and deletions in the process. And then the match would continue. If we find a sufficiently good match, we'd stop. Or if the match is not good, then we will stop. Then that wouldn't be a match for the reads onto the reference in the alignment step. So here, what we are looking at in this table, these three columns corresponds to the default seed length of 24 in BWA. And these three columns corresponds to the top 10 differentially expressed genes when we reduce the seed length from 24 to 0. So, and in their original paper, Lee and Durbin had documented that a smaller seed length generally lead to more accurate results, but would take more computational time. So here, these are the default parameters, seed length of 24, the top 10 differentially expressed genes. So what we are looking here are the p-values. So smaller p-value is more significant in terms of the differential expression results. And these color, the more significant p-value would have a lighter shade of blue. And the orange color here are the comparing the rank of the top 10 differentially expressed genes. So for example, for the default parameter, the top differentially expressed genes for this particular drug perturbation is VIM. And even when we reduce the seed length to 0, VIM remains the top differentially expressed genes. But when we go down the list, we see some more changes. So for example, RCN1, it's the top 4 differentially expressed genes when the seed length is 24, but it would go down to top 5 when we reduce the seed length. And a more dramatic example is SLC30A4. So this one, it's on the top 10 list, but if we change the seed length to 0, it will reduce the ranking by 14 positions and will not be on the top 10 list at all. Conversely, SLC9A7 is on the top 10 list when the seed length is 0, but it's not on the list when the seed length is 24. So because typically biological interpretations are made using the top differentially expressed genes. And these, so what we illustrated here is by changing the input alignment parameters, we could change the list of differentially expressed genes, therefore could potentially change the biological interpretation of the results. So next, we are going to present some of the tools that we developed in the past few years that focus on optimization of computational intensive tasks, but in different ways. So the first work here is we have a framework called hot start containers. A lot of the bioinformatics workflows have an initialization step. So if you launch the containers again, all the steps would be, if you we do the workflow, all the steps would be repeated again. But the idea of hot start containers is we will save the intermediate step, the results from the intermediate step, so that we don't have to repeat all the steps again. So in the example of our NA-seq workflow, so oftentimes there is an indexing step using the reference sequence. So we showed in this work published in eScience 2018 that hot start containers can save us execution time on different cloud platforms. And next, it's a holistic optimization of our NA-seq workflow for multi-threaded environments. So these two papers are highlighted in bold because both of them use the UMI RNA-seq dataset from Mount Sinai that I have described. So in this paper published in Bioinformatics 2019, as we took the workflow, the SOP from Mount Sinai that took 29.5 hours, and we optimized all the steps in the analysis, not the alignment step, but also because the alignment step is the most compute intensive step, we also optimized the IO bound steps. And we cut down the running, and we added, we improved the implementation, we added multi-threading, and we improved the algorithm, and we improved the running time from 29.5 hours to 3.5 hours using AWS EC2 instances. And we also optimize in terms of cloud architecture. So we have been looking at serverless computing tools. And in this effort, we leverage serverless computing tool. It's more like a proof of concept to show it would work for bioinformatics application. In this particular paper, we use a pairwise protein sequence comparison as an example. And this work was published in the ACM BCB workshop there, 2019. And we are going to focus more on talking about how we used optimize the cloud architecture for interactive analysis using for our NA-seq analysis here. And this work, we have updated our preprints on bioRxiv, and this currently under journal revision. So we'll focus on that in the rest of the talk. So back to our challenges slide. So here we are going to address the computational intensive challenge by performing holistic optimization that accounts for all steps in the workflow, and also by optimizing the cloud architecture as well. So a little bit more background on serverless computing, cloud computing. So this is, we see a nice big cloud on the screen. So serverless computing is also known as FaaS, function as a service. And there are many advantages of serverless cloud computing. There is reduced setup, high availability. We pay only for the actual runtime. So most bioinformatics tools on the cloud are deployed on virtual machines that we need to configure the machine, the VM, and then we are charged for the amount of time that the VM is up, but not necessarily when the application, when the job is being run. So in serverless cloud computing, the setup is simpler, and then we only pay for the actual runtime, and also if we need to scale, that's also we don't need to manage that ourselves. So it's kind of an abstraction of VMs. But it also, there is nothing perfect in this world. It also comes with some limitations. So there are limitations in terms of memory available, runtime of each serverless function, the disk capacity, and the supported language, and the code size that we can upload for the serverless platform. So serverless functions are available in most major cloud providers. For example, on Amazon Web Services, we have the AWS Lambda. On Google Cloud, they have the Google Cloud functions. On Azure, there are the Azure functions as well. So this is the hybrid cloud architecture that we developed for the UMI RNA-seq workflow. By the way, our optimization also works for regular RNA-seq, not just UMI RNA-seq data and workflows. So there are four key components in our architecture. So these are the clients that perform the non-compute bound steps. These are EC2 instances, or it can be your local host. And we have these serverless, the second component would be the serverless functions that essentially allow us to do the alignment step in a very highly parallel manner. And then we also have the storage buckets that would store our input file, output files, and intermediate files as well. And we also use a message queue to alert us when we can call the serverless function instances to perform the alignment using BWA, and when also let us know when the results are being done. So in this architecture, we start with putting all the input FASTQ files in an S3 bucket. And the split step, in the split step, we take the 640 million reads and chop them up into smaller chunks so that each of the smaller chunk, the alignment step, can be performed on a serverless function. And then we put these smaller chunks, the shards, back into the storage bucket. And so these storage bucket are the S3 on AWS or the Google storage bucket on Google Cloud. So our workflow works on both AWS and Google Cloud platform. And then we would send a message in the message queue to let us know, hey, we have this particular shard ready that alignment can start. So we would start 17,000 serverless instances that each of these serverless instances would perform the BWA alignment. And then we collect the results. Again, the optimization we do is not just by optimizing the cloud architecture. We also optimize the algorithm and the file transfer to reduce the size of the data that needs to be transferred between the functions and the different buckets and the different instances. So we put our hash values of the output back in the storage bucket. And then we have a merge step on another EC2 instance, on another client. This client can be an EC2 instance, or it could be your local desktop computer. And at the end of the day, we would get the table of counts. So results of scalability, how we have improved performance. So what we are looking at is this column has the different UMI RNA-seq implementation. And this is the time needed for the alignment step. And this column represents the total amount of running time for all steps in the workflow. So the formats are in hour, minute, and second. So the original workflow provided to us, as I said before, takes 29.5 hours on an EC2 M446 large instance. And the alignment step takes about roughly 19.5 hours. And the optimized workflow that was previously published in 2019 in the bioinformatics application, in the bioinformatics journal, we optimize it down to 3.5 hours for the entire workflow, and the alignment takes roughly 2.5 hours. And that this also used EC2 instances and 16 threads. So This work if we use EC2 instances as a cloud client and using AWS Lambda functions. The whole workflow would take roughly 6 minutes, and the alignment step can be done in a little bit more than 1 minute. So this is 142-fold speed up compared to our previous publication. And for the total workflow, it's a 35-fold speed up compared to our previous paper. And when comparing to the original original alignment step, original workflow in Python, it's a 1,100-fold speed up, and the total workflow represents a 290-fold speed up here. And if we use a local client is your desktop or laptop, that the alignment step can be done in roughly 2.5 minutes, and the total runtime is about 19 minutes. So in each of these experiments, we repeated our empirical experiments 5 times. And what we are showing here is the median runtime. So back to our challenge slide. So now we have talked about the holistic optimization that accounts for all steps in the workflow and the system's architecture. Next, we are going to talk about the next challenge, which is the technical expertise required to run these workflows on the cloud. So we also provide a graphical front end that simplifies configuration of cloud services. And so in terms of accessibility, we extended the BWB, the BioDepot-workflow-builder framework to support serverless workflows for both AWS and Google Cloud Platform. So what we are seeing here is an implementation of our hybrid cloud architecture that presented a few slides ago. So here it's the setup step that we simplify. So for the user, all the user needs to provide the credentials on their cloud AWS account or the Google Cloud account, and we would take care of the configuration for you. And here, this is the split step, and then this is the function instances for the alignment step, and this is the merge step, and then finally, this is the differential expression genes identification. So at this point, I can also pause my go back to the, let me see, go back to the demo here. So this is, how come the window have the, oh, hey, here, I think all I need is here. So this is all the input parameters. So the user provide the AWS credentials in a mounted directory, and then specify some like input parameters as well for doing the alignment. And then clicks the start of a button. I'm not going to do that again. I have previously run it. And then at the end, we will get the top differentially expressed genes here. Let me see if I could move the windows around. I think the zoom window is hiding here some of my other windows. Well, see if I could. I don't know where that window is. I would try to find it in the interest of time. It's already 5:15. And so this is, let's get back to our presentation here. So a quick recap in terms of scalability, we have reduced the execution time from the original 29.5 hours to roughly 6 minutes, such that scientists can explore the impact of different parameters, including parameters in the very computational intensive step, alignment step, on analytical results. And we have shown with real data sets how changing the input alignment parameters can change the genes, the list of genes that we get at the end of the analysis. In terms of accessibility, we provide a graphical user interface so that biomedical scientists can deploy workflows on the cloud and leverage serverless computing platforms with a point and click, drag and drop interface without needing any command line tools. So our approach has potential to transform our NA-seq analysis to be interactive and efficient. When you can complete your workflow in 6 minutes instead of more than a day, you could afford to change many input parameters, experiment with different methods, different parameters, different tools to see how that could impact the results. And the other way that our tools can transform analysis is we would make the life of bioinformaticians easier so that bioinformaticians can focus on the intellectually interesting jobs of designing workflows and new tools and new methods to improve the quality of results rather than rerunning command line tools. Okay. And so, so these are things that projects that we have written up. So what else are we currently working on? So we also put a lot of effort in benchmarking bioinformatics workflows to optimize cloud resources. Not only we do the benchmarking, we also develop tools to do the benchmarking. Our goal is that we need to understand the resource utilization characteristics of all the steps in the workflow, not just the alignment step, in order to, so that we could, we know how to optimize. We can explore different ways to do our holistic optimization, not just the step that everybody is optimizing. We want to optimize all the steps in the analysis as well. And we are very interested in to expand the applications of our work via collaborative projects and build a user community. And so we are adding more workflows, more tools to our to the BWB. And one of the example is that we are going to support integrative cancer genomics workflows. And we are going to do a pilot use studies in early summer. So we welcome collaboration, ideas, questions, feedback, and if you would like, if you're interested to participate in the pilot use studies, please send me an email.
Next question is, how do you end up deciding serverless instances number? For example, 1,752.
Great question. So we did a lot of empirical experiments to get down to 6 minutes. And also so there are a couple of considerations. One is we need to make the groups of sequences small enough that we can fit in the memory, in the runtime, in the disk space of each function instance. And within that multiple choices, we want to make it as efficient as possible.